Imagine an invisible, tireless army always on guard inside you. A force so sophisticated it actually learns from every encounter, remembers its adversaries, and constantly adapts its strategies. Today, we're taking a deep dive into the structure and function of the immune system. And for you, our dedicated learner, this isn't just about, memorizing a list of biological facts. It's really about unlocking a profound understanding of the incredible, intricate defense network that tirelessly keeps you healthy every single day. Our goal here is to offer you a bit of a shortcut to being truly well-informed about your own body's amazing capabilities, hopefully packed with some surprising facts and insights that might just spark some genuine aha moments. We've pulled together an incredible stack of sources for this deep dive, everything from intricate cellular breakdowns to broad structural overviews, even some historical perspectives and cutting-edge research. Our mission today is to unpack this complex system, revealing the fascinating roles each organ and specialized cell plays. And crucially, to show you how it all works together, in perfect concert to protect you from all sorts of unseen threats. So let's jump right in. Okay, let's unpack this then. When we talk about the immune system, it sounds like, well, maybe a single thing, like one shield. But what exactly are we talking about? How vast is this system, really?
That's a perfect place to start, because you're right, it sounds simple, but it's anything but. At its core, the immune system is this highly complex and honestly extraordinarily coordinated network. We're talking specialized cells, tissues, organs, all spread throughout your entire body. Its fundamental concern, its whole reason for being, is managing your body's defense reactions. It's relentlessly dedicated to maintaining your body's integrity. How? By recognizing and then eliminating a whole range of threats. And these threats, they aren't just the external invaders you might think of first, like, say, bacteria and viruses. It's also constantly patrolling for internally derived threats, things like rogue cells that have malignant potential. So essentially, it's protecting you from both outside invaders and internal elements that could turn against you. It's like this internal security force that literally never sleeps.
Okay, so definitely not just a single shield. It's a whole interconnected security system constantly scanning. And it's not just one type of defense strategy either, is it? I remember reading about these two big sort of fundamental categories that define how our body fights things off, almost like different branches.
Precisely. Yeah, you can absolutely think of it as having two main pillars of defense. Each one has its own unique strengths, its own, you could say, operational philosophy, even its own history in evolutionary terms. First, you've got natural or innate immunity. This is your body's pre-existing, born-with defense system. It's always on, always ready to respond to a general kind of threat. Its key characteristic. It's antigen non-specific. Now, what that means is it doesn't really need to know the specific identity of the pathogen. It doesn't care if it's a specific cold virus versus a strep bacterium. It just reacts to anything it broadly recognizes as a generic danger pattern, something that signals non-self. This system acts as your immediate, your very first line of defense. It's ready to respond instantly. No prior exposure needed. It's your rapid response unit. And crucially, its efficiency doesn't get better with previous encounters. It doesn't learn in that sense. Think of it like a general alarm system in a building. It goes off whenever there's an intruder, regardless of who they are or if they've broken in before. It just senses an intrusion and triggers a broad, rapid response.
That makes a lot of sense. The innate system is the immediate general alarm. Quick, but not specific. But that other pillar, adaptive immunity, that sounds like it's a bit more targeted, maybe. More sophisticated. Something that actually learns over time.
That's exactly right. And that's where the truly remarkable evolution of our immune system really shines. The adaptive or acquired immunity system, it's far more sophisticated. It operates on a completely different level of precision. It responds in what we call an antigen-specific manner, meaning it meticulously identifies and targets the precise, unique threat it encounters. It tailors its attack specifically to that particular pathogen. And what's truly remarkable about this system, and here's maybe one of those aha moments, is its ability to retain a profound memory of the threat. This means if you encounter the exact same pathogen again in the future, your adaptive system doesn't start from scratch. No way. Instead, it can mount an accelerated and significantly heightened protective response. This is precisely why you often don't get the same illness twice. Or if you do, it's usually a much milder case. Your body remembers. And it's also the fundamental principle behind how vaccines work. They essentially introduce a harmless mugshot of a pathogen or a piece of it to your adaptive immune system. This allows it to develop that crucial memory without you ever having to experience the full-blown disease.
Right. So it's like a highly trained special forces unit. It learns the enemy's weaknesses, gathers intelligence, remembers the specifics for future missions, and becomes stronger, faster, more efficient each time it encounters that same enemy. That's incredibly clever. So how do these two systems, the innate and the adaptive, how do they interact? Do they just work independently, or is there collaboration?
Oh, they absolutely collaborate. And that synergy, that teamwork, is what makes our overall immune system so incredibly robust. You can think of it like a carefully choreographed relay race, maybe, but one with a constant communication between the runners. Innate immunity provides that immediate, broad protection. It's like the first responder arriving on the scene. It holds the line. And importantly, it buys valuable time. While it's doing that, it's simultaneously signaling to the adaptive system. It's passing on crucial intelligence about the nature of the threat it's encountering. This signaling allows the adaptive system to then kick in to precisely identify the specific threat, learn from it, and then provide that long-lasting, highly targeted defense we talked about. It's a truly beautiful and incredibly effective example of biological synergy. The quick general response really sets the stage perfectly for the powerful, precise, and persistent defense later on.
Okay, so we have these two incredible types of immunity working together, the immediate innate, the specific adaptive with its memory. Where does this whole complex defense force actually get its start? And where do these critical interactions happen? I mean, I imagine the immune system isn't just, floating around randomly in our bodies. It must be highly organized within specific tissues and organs, right? These lymphoid tissues.
Absolutely right. It's highly organized. These organs are collectively known as lymphoid or immune tissues and organs. And we broadly categorize them into two groups: primary or central lymphoid organs and secondary or peripheral lymphoid organs. This categorization is based purely on their function, on what they do. You can think of the primary organs as the boot camps or maybe the schools. This is where the immune cells are born and receive their initial fundamental training and education, getting them ready. The secondary organs then are more like the forward operating bases or the battlefields. These are the places where those mature, trained cells actually encounter the threats and launch their coordinated responses. So focusing first on those primary lymphoid organs. These are the major sites for a really crucial process called lymphopoiesis, which literally just means lymphocyte creation. It's where precursor lymphocytes proliferate, they develop, and they differentiate into immunologically competent cells, ready to function. They're where the soldiers of your immune system are essentially recruited, born, and get that vital initial training before they're deployed out into the wider body.
Okay, let's dive into the first of these primary organs. And this is where it gets really interesting for me, the bone marrow. We often think of bone marrow mainly for, making blood cells, red blood cells mostly, but it's absolutely central to immunity too. It sounds like the real starting point for so many of these amazing cells. How foundational is it?
Well, it's not just foundational, it's the absolute origin point. In mammals, the bone marrow is the primary site of what we call hematopoiesis. That means the formation of all blood cells, period. Red cells, platelets, and yes, the entire spectrum of immune system cells. Precursors for both the lymphoid cells, like your B and T cells, and the myeloid cells, which include a whole host of those immediate responders, they all derive from these special cells called self-regenerating hematopoietic stem cells, or HSCs. And these HSCs are found right there in the bone marrow. They're truly the mother cells that can essentially become any type of blood or immune cell. So specifically, the genesis, the birth, and the maturation of B cells, those crucial antibody-producing cells, primarily takes place right here in the bone marrow after birth. So its key functions are really multifaceted. First, primary site for forming all blood cells, the entire immune army. Second, it's a key site for lymphocyte development, particularly for B cells. They mature here, and they undergo these incredibly complex gene rearrangement processes. This is essentially how they learn to recognize a vast, almost unimaginable repertoire of different potential threats. It's like giving them billions of unique keys designed to match countless potential locks on invaders. Third, it serves as a vital stem cell reservoir, constantly maintaining that supply of those critical HSCs. And finally, and this is important, in direct response to things like infections or injury, the bone marrow can dynamically ramp up production of specific types of immune cells. It custom tailors the output to bolster the immune response exactly when your body is under attack. If you ever look at a diagram of the differentiation tree starting from an HSC, it's honestly astounding to see how one single type of foundational stem cell gives rise to such a vast and specialized array of immune defenders, each one perfectly designed for its specific role.
That really does sound like the ultimate supply depot and recruitment center for the entire immune force. Just incredible diversity coming from one source. And then you mentioned some of those cells, particularly the T cells. They have to go off to a very special school, the thymus, for their more specialized education. What makes this thymus school so rigorous? I heard it's pretty intense.
It truly is rigorous, and it's an absolutely vital school.
The thymus is quite unique. It's actually the first lymphoid organ to develop in the body, starting even before birth. And it's the critical site where T lymphocytes, or T cells, mature. It plays this vital, almost philosophical role in educating these emerging T lymphocytes. Educating them to do what? To accurately discriminate between self components, things that belong to your own body, and non-self foreign invaders, things that don't belong. Now, this education process, it involves an incredibly rigorous selection process. We call it positive and negative selection. During positive selection, any T cells that can't properly recognize antigens presented by your own body's major histocompatibility complex, or MHC molecules, well, they're eliminated. They simply wouldn't be functional. But here's the really surprising part, maybe another aha moment about the thymus. During negative selection, T cells that react too strongly with your own self-antigens are also eliminated. This is absolutely critical because if they weren't removed, these rogue T cells could potentially launch devastating attacks against your own healthy tissues.
This is what leads to autoimmune diseases. In fact, the process is so rigorous, it's estimated that only about 2%, just 2% of the T cells that enter the thymus actually graduate and are allowed to leave. The other 98%, they're eliminated right there. It really showcases just how critical this self-recognition, this tolerance is for preventing, friendly fire. It's an incredibly meticulous quality control system, absolutely essential for preventing your own immune system from turning against your healthy tissues.
Wow, only 2%. That puts a whole new perspective on how finely tuned our body's defenses really are. That failure rate is astonishing. So once these elite 2% of T cells are educated and validated, where do they head off to? They're ready for action, presumably.
Exactly. Once they're educated, validated, they've passed the test, these mature T cells are released into the bloodstream. From there, they migrate to the secondary lymphoid organs, places like the lymph nodes and the spleen, fully ready for action, ready to encounter antigens. The thymus also plays a slightly less direct but still important role in immune memory. It contributes to the formation of memory T cells during an initial immune response. And these memory cells then circulate, constantly on alert, capable of responding much more rapidly and effectively if they encounter that same pathogen again down the line. The implications of this thymic education are just profound. If this critical process goes awry, if those 2% aren't selected perfectly, well, it really raises important questions about the origins of conditions like autoimmune diseases, where that non-self discrimination breaks down. It's an elegant system, but the stakes are incredibly high. Okay, so once these immune cells, both B and T cells, are mature and have completed their vital education in those primary organs, they're not just floating around aimlessly, they move strategically to the secondary lymphoid organs. These are essentially the meeting points. You could call them the action zones, or those forward operating bases we mentioned. This is where mature lymphocytes encounter foreign antigens, where they become activated, and where they then differentiate into the powerful effector cells that carry out the actual immune response, engaging directly with threats.
Right, the lymph nodes. We've all felt them, haven't we? Swollen in our neck or maybe under the armpits when we're sick. It's often a really clear sign that our body is fighting something off. What exactly are they doing inside us that causes that noticeable swelling? And how do they function as these meeting points?
They are truly fascinating little structures. They're small, encapsulated, kind of bean-shaped, and they're strategically placed all throughout your body, almost like sentry posts along your body's internal roadways or lymphatic vessels. Their primary function is to act as highly sophisticated filters for lymph. Lymph is that clear fluid that continuously bathes your tissues, picks up waste products, and critically carries immune cells and any foreign particles it encounters. So you can imagine them as a series of highly efficient security checkpoints. Lymph nodes are meticulously designed to basically concentrate any foreign antigens, those fragments or marchers and invaders, onto specialized cells called antigen-presenting cells, or APCs. These APCs then present these antigens to the lymphocytes residing in the node, kicking off a precise, targeted immune response. So when you feel your lymph nodes swelling up, that's often a sign that these checkpoints are incredibly active. They're buzzing with activity. They're ramping up the production of immune cells because they've detected a significant threat that needs dealing with. Inside the node, the lymph fluid, potentially carrying threats, enters through multiple incoming vessels. It then slowly percolates to this dense meshwork structure inside the node. And as it does, it's meticulously scrutinized by a huge army of macrophages and lymphocytes that are constantly patrolling, ready to mount a response. Any detected threats are immediately brought to the attention of the specialized units, the T and B cells. The cleaned lymph then leaves the node via outgoing vessels. And it's quite stringent. Lymph usually has to pass through several nodes in a chain before it's considered clean. This ensures incredibly thorough screening and really maximizes the chance of catching any invader trying to sneak through.
That visual of security checkpoints really helps clarify their role. filtering the lymph, activating responses. Okay, what about the spleen then? It's often kind of overlooked. Maybe people don't fully understand its role, but it sounds vital, and it's doing something a bit different from the lymph nodes, filtering blood instead.
Absolutely. The spleen is actually the largest lymphoid organ in your body. And yes, it's distinct from lymph nodes in that crucial way. Lymph nodes filter lymph. The spleen acts as a crucial filter for your blood. It's a major site for clearing out what we call opsonized particles. These are essentially pathogens, like bacteria, that have been tagged for destruction by antibodies. The spleen pulls them out of circulation. And it's also a key location for producing antibodies directly against antigens that are found circulating in the bloodstream. So if a nasty bacterial infection manages to get into your blood, causing sepsis, for example, the spleen is a really critical frontline defender. Structurally, the spleen is kind of divided into two main types of tissue. You've got the white pulp. This is the lymphoid tissue, containing distinct T cell and B cell areas where active immune responses happen. It's basically the spleen's own immune headquarters. Then there's the red pulp. This is a complex network of blood-filled sinusoids, and it's packed with macrophages and red blood cells. The red pulp is primarily involved in filtering the blood itself. And beyond its huge immune functions, the spleen also plays another critical role, the destruction of old and damaged red blood cells. It helps keep your blood healthy and efficient. It's also a critical production site for activated lymphocytes, which are then delivered back into the blood to help fight systemic infections throughout the body. So the key takeaway for the spleen: it's an essential blood filter and a significant antibody-forming organ. It's tirelessly working to keep your circulating blood free of threats and debris.
Fascinating. So lymph nodes check the fluid draining from tissues. The spleen filters the blood itself. But then there's this other category, MLUU mucosa-associated lymphoid tissue. That sounds like it's protecting the very front lines of defense, literally where invaders first try to get in, like our gut, our airways.
Precisely. MALT represents a truly ingenious and strategically vital defense system. It's basically a collection of non-encapsulated dispersed aggregates of lymphoid cells. So unlike the distinct encapsulated structures like lymph nodes or the spleen, MALT is more diffused. It's spread throughout your body's mucosal surfaces. And these surfaces are the main passages where microorganisms gain entry into the body. We're talking the alimentary or digestive tract, the respiratory tract, and the urogenital tract. Think of these as the body's border zones, right? They're constantly exposed to the outside world. And MALT is strategically positioned right there to guard them. Examples of MALT that people might recognize include gut-associated lymphoid tissue, or GALT. This includes well-known structures like Peyer's patches, which are found in the lower part of your small intestine, your tonsils, and various other lymphoid tissue accumulations all throughout the intestinal wall. Within Peyer's patches, for instance, there are these specialized cells called M cells. They act almost like tiny tasters or samplers.
They actively take up particles from the gut lumen and deliver them directly to macrophages and other APCs lying just underneath. These then present any antigens to lymphocytes. It's essentially sampling the gut environment constantly for potential threats you might have ingested. Similarly, you have bronchial-associated lymphoid tissue, or BALT, found in the respiratory system, doing a similar protective job for the airways. MALT contains this large, dynamic population of antibody-producing plasma cells ready to go, and it facilitates crucial interactions among circulating immune cells right there at the surface. This allows for immediate, localized immune responses right at those critical points of entry. It really highlights how the immune system isn't just some internal fortress. It has specialized sentinels deployed directly at the body's most vulnerable interfaces with the external world, ready to intercept invaders before they can truly establish a foothold. It's constant, active surveillance right at the gates. Okay, so now that we've explored the command centers and training grounds, the primary and secondary lymphoid organs where our immune cells are born, educated, and deployed, let's talk about the specialized soldiers themselves. Let's get into the incredible cells of the immune system. And it's really vital to remember all of these cells, no matter how incredibly specialized they become, they all originate from those self-regenerating hematopoietic stem cells we discussed earlier. Those HSCs in the bone marrow and also in the fetal liver during development. From that single starting point, they embark on these remarkable journeys of differentiation, specializing along either the myeloid pathway or the lymphoid pathway. And this is what ultimately leads to the incredible diversity of immune cells we find protecting us.
Right, and what's truly fascinating here, I think, is how incredibly specialized these cells are. Each one seems to have a unique, really defined role in the body's complex defense strategy. It's definitely not just a generic army. It feels more like a highly compartmentalized, skilled force with lots of different units, each expert in its own task. Let's start with the lymphocytes. They're often considered the elite responders, aren't they? Maybe the true sharpshooters of the immune system.
They absolutely are the elite responders. Lymphocytes are the major cellular players directly responsible for your specific immunological responses. What makes them unique is their ability to interact precisely and specifically with antigenic substances. They can distinguish one invader from another with incredible accuracy. And as we touched on, they are the crucial contributors to immune memory, that long-term protection. They are absolutely tireless in their duties too. They continuously circulate between your blood, your lymph fluid, the lymphoid organs, and various tissues throughout the body. They're always on patrol, always ready to detect and respond to any signs of trouble. Now, within this category of lymphocytes, we first have the B lymphocytes, or B cells. As we said, they primarily mature right there in the bone marrow. Their key function is really quite remarkable. They express a unique receptor on their surface, the B cell receptor, or BCR. You can think of this BCR as being like a highly specific antenna. It's shaped perfectly to recognize and bind to one specific type of invading molecule, an antigen. Their primary role, once they get activated by actually encountering their specific antigen, is to differentiate into plasma cells. And plasma cells are literally biological factories. Their job is to secrete massive amounts of immunoglobulins, which we commonly know as antibodies. These antibodies are like the smart bombs of the humoral or antibody-mediated immune system. They provide incredibly powerful and targeted defense, especially against extracellular threats like bacteria and viruses floating in your body fluids.
So they're the producers of these precision weapons, the antibodies. Do B cells have any other roles besides making antibodies eventually?
Yes, they actually have a crucial dual role. B cells can also function as highly efficient antigen-presenting cells, or APCs, particularly for T lymphocytes. They can capture a pathogen, process it internally, and then display fragments of it on their surface using MHC molecules. They basically show off what they've encountered to T cells, effectively helping to rally the T cell part of the response. And importantly, after that initial encounter with an antigen, a population of long-lived B memory cells gets selected for survival. These memory cells remain vigilant, sometimes for decades, enabling that much faster and significantly enhanced antibody response if you re-encounter the same antigen later. This is critical for long-term immunity. It's also worth noting, as an interesting detail, that these antibody-producing plasma cells, once they're fully differentiated, they typically don't migrate to the site of infection themselves. They tend to stay put in lymphoid tissues, like lymph nodes or bone marrow, and just keep churning out antibodies that circulate throughout the body. And our sources also note that plasma cells themselves don't really react against things like transplants or cancer cells. Those threats primarily engage T cell responses, which really highlights the precise division of labor within the immune system's grand design. Okay, then we have the T lymphocytes, or T cells. Now remember, they originate in the bone marrow like B cells, but they must migrate to the thymus to fully mature and undergo that critical education process we talked about earlier, checking for self-reactivity. T cells express their own specific antigen-binding molecule, called the T cell receptor, or TCR, on their membrane. Crucially, and this is a really key difference from B cells, TCRs only recognize antigens when they presented to them by major histocompatibility complex, or MHC, molecules on the surface of other cells, specifically antigen-presenting cells or infected body cells. Think of it this way maybe. B cells have those BCR antennae that can directly grab onto invaders floating freely in body fluids. T cells, however, are more like highly specialized detectives. They need a waiter, that's the APC or infected cell, to present a specific sample of the invader on a special plate, that's the MHC molecule, before they can recognize it. They generally can't just see the antigen floating freely. T cells form the absolute core of the cell-mediated immune system. They're the ones that directly engage with infected or abnormal cells in your body.
That analogy of the waiter and the plate really helps clarify how T cells operate so differently from B cells. They need that specific presentation. And within T cells, you mentioned there are different subsets, right? Different kinds of T cells.
Absolutely. T cells aren't just one monolithic group. They're categorized into major subsets, each with highly specialized roles. And we can often identify them by different cluster of differentiation or CD markers on their surface. First, there are the T helper, or T8 cells. They're often identified by the CD4 marker. These cells are truly the coordinators, or maybe the conductors of the immune orchestra. They don't typically kill invaders directly themselves,
but they're absolutely essential for activating and directing the effector functions of other immune cells, B cells, other T cells, natural killer cells, even macrophages. They achieve this vital coordination through direct cell-to-cell contact and, very importantly, by secreting those soluble signaling molecules called cytokines. Remember, cytokines are like the immune system's language, transmitting instructions, alerts, and commands between cells. Some T helper cells, for example, specialize in activating macrophages and other T cells to fight invaders that live inside our own cells, like viruses or certain bacteria. We call these Th1 cells. Others, called Th2 cells, are masters at boosting B cell activity, helping them to switch antibody types and churn out huge amounts of antibodies, particularly important for fighting extracellular parasites. And more recently, another subset called Th17 cells has been identified. They produce a cytokine called IL-17 and are very pro-inflammatory, playing roles in fighting certain infections, but also implicated in the pathogenesis of some autoimmune diseases. So T helper cells are the managers, the directors. Next, we have the T cytotoxic, or TC cells. These are usually identified by the CD8 marker. Now, these are the killer cells of the adaptive immune system. Their role is direct and decisive. They directly kill target cells. What kind of target cells? Your own body cells that have become infected with a virus, or perhaps cells that have turned cancerous. They recognize these compromised cells because the infected or cancerous cell displays abnormal fragments, bits of virus or tumor proteins on their surface, presented via MHC class I molecules. The TCS cell's TCR locks onto this complex. These are the cells that literally move to the site of infection or tumor growth and directly engage the threats, eliminating those compromised cells before they can cause more harm. And as we noted, our sources specifically highlight that these killer T cells are the ones that react against foreign transplants and are absolutely crucial in the body's ongoing battle against cancer cells. Then there's another really important group, the regulatory T, or TR cells, sometimes called Tregs. You can think of these as the peacekeepers or the balancers of the immune system. Their crucial role is to actually downregulate or suppress immune responses. They also regulate anti-self reactions. This is absolutely essential. Why? To prevent excessive inflammation once a threat has been cleared. You don't want the immune response raging out of control. And critically, they help prevent autoimmune diseases by keeping those potentially self-reactive T cells in check. They effectively tell the immune system, okay, stand down when the threat is neutralized or if there's a risk of the immune response causing too much collateral damage to your own healthy tissues. And just like B cells, T cells also form long-lived memory populations after an initial antigen encounter. These T memory cells provide that accelerated, enhanced secondary response, giving you lasting protection.
Okay, moving on. These next cells have such a direct, almost, aggressive name, natural killer cells, NK cells. What makes them so special? And why are they considered natural? Does that mean they're part of the innate system?
It's a great question, and yes, their name perfectly describes their function, and they are indeed considered part of the innate immune system, though they are lymphocytes. Natural killer, or NK cells, are non-phagocytic lymphocytes. That means they don't engulf other cells or particles like macrophages do. They actually account for a significant chunk, maybe up to 15% of the lymphocytes circulating in your blood. Their special innate role is in directly killing certain target cells, specifically virus-infected cells and malignant or tumor cells. What makes their activity natural, or non-immune, as the term implies, is that it does not require any prior antigenic contact or specific training through antigen presentation, unlike the adaptive T and B cells. They are always ready to act, part of your innate immunity, acting as a kind of rapid response surveillance and cleanup crew for abnormal cells. And a really fascinating aspect is their activation mechanism. They're often activated when there is no inactivation signal coming from MHC class antigens on a target cell. Now here's an interesting point, maybe another aha moment. Many virus-infected cells and also many tumor cells strategically try to hide from the adaptive immune system, specifically from those killer T cells. How? By downregulating or removing their MHC class I molecules from their surface. It's like they're trying to take off their identification badge to avoid being recognized. NK cells are cleverly designed to recognize this absence of the normal self-markers, this missing ID badge. When an NK cell detects a cell lacking the expected MHC-I signal, it interprets that as a sign of danger, either infection or malignancy, and moves in to eliminate the threat. This makes them a crucial early warning and response system. They're particularly effective against threats that try to evade the adaptive immune system by hiding their identity. They're your immediate
intelligent surveillance systems specifically looking for rogue cells.
That's incredibly clever. So they spot the cells that are trying not to be seen by the other killer cells. Okay, so if lymphocytes like T cells and B cells are the precision forces, the highly specialized units with specific targets, and NK cells are the specialized innate killers, what about the cells that are more like the general infantry, the cleanup crew, the first responders that just engulf and eat things, the phagocytes?
Those would indeed be your phagocytic cells. And they are exactly as you described. Their main job is to ingest or eat extracellular particulate material. This includes whole pathogenic microorganisms like bacteria, cellular debris, things like that. If your body's mechanical defenses, like your skin, get breached so you get a cut, these phagocytic cells form a crucial immediate barrier, rushing to the scene to deal with invaders. They are absolutely fundamental components of your innate immunity, acting as those immediate responders right at the site of intrusion or infection. Now, within the phagocytic cells, we have a group often referred to as granulocytes, or sometimes polymorphonuclear leukocytes, because they're funny-shaped nuclei. These cells are characterized by their distinctly segmented or lobulated nuclei and the presence of prominent granules in their cytoplasm, which gives them their name. Among them, neutrophils are incredibly important. They are relatively short-lived, but they are highly effective phagocytes. They circulate in your blood in huge numbers, and they can rapidly invade tissues to confront infection when signaled. They are absolutely crucial in what we call pyogenic inflammation. That's inflammation characterized by the formation of pus. Pus is largely made up of dead neutrophils that have done their job fighting bacteria. Neutrophils are the most abundant type of white blood cell in your circulation, and they are very often the very first responders to a bacterial infection, arriving quickly and in large numbers to engulf and destroy the bacteria. Then you have eosinophils. These are phagocytic, but perhaps to a more limited degree than neutrophils. They seem to play a particularly important role in defense against parasitic infections, like parasitic worms. They release granule contents that can damage these larger parasites. Eosinophils are also commonly found in allergic inflammation, contributing to symptoms in conditions like asthma or hay fever. And finally, within the granulocytes, there are basophils. These are actually non-phagocytic. Instead of engulfing things, they release potent pharmacologically active substances like histamine and heparin from those large granules in their cytoplasm. They play a major role in certain allergic responses and in inflammation, contributing to things like blood vessel dilation and attracting other immune cells. Okay, another crucial group of phagocytic cells are the monocytes and macrophages. These two are closely related. Monocytes are relatively large leukocytes that circulate in your bloodstream for a day or so. But once they leave the blood and migrate into various tissues throughout your body, they differentiate and mature into macrophages. These macrophages are mature, long-lived, tissue-resident phagocytic cells. You find them strategically positioned in various areas of blood filtration and high immune activity. Think liver sinusoids, the filtering units in the kidneys, the air sacs, alveoli in the lungs, and of course within the lymph nodes in the spleen. Their primary function is definitely to kill microbes by engulfing them whole phagocytosis and then breaking them down using powerful enzymes and low pH inside specialized internal compartments called phagolysosomes. But, crucially, beyond their impressive eating function, macrophages also act as very important antigen-presenting cells, APCs. Like dendritic cells, they can show off bits of the invaders they've eaten to activate T cells. And they also secrete a wide range of cytokines, effectively communicating vital information and instructions to recruit and direct other immune cells. They are truly the big eaters and also vital communicators of the immune system, acting as both a sanitation crew and important intelligence gatherers. And then we have the dendritic cells, or DCs. These are also bone marrow-derived cells. They're characterized by their long, narrow, almost tree-like processes dendrites, which give them their name. These processes make them incredibly efficient at contacting and sampling their environment for foreign materials. Dendritic cells are arguably the most potent, the most professional antigen-presenting cells in the immune system. They are especially critical for presenting antigens to naive T cells that haven't encountered antigen before during the primary immune response. They are absolutely key for initiating that crucial adaptive immunity. You can really think of them as the scouts or the messengers of the immune system. They are strategically present in tissues that interface with the external environment, like the skin, where they're specifically called Langerhans cells, and also in the mucous membranes lining your gut and airways. They act as alert sentinels right at these surfaces. Once they capture antigens from an invading pathogen, they don't just stay put. They undergo maturation, and then they actively migrate from the periphery, carrying that vital intelligence, the mugshot of the invader,
you could say to the local lymph nodes. And it's there, in the lymph nodes, that they present these antigens to the naive T cells, activating the specific elite forces of the adaptive immune response. There's even a specific type called follicular dendritic cells, or FDCs. These are a bit different. They're found in the B cell areas, follicles and germinal centers, within lymphoid organs like lymph nodes and the spleen. They specialize in trapping and displaying antigen-antibody complexes, which is important for B cell activation and memory development. So dendritic cells are truly master orchestrators, bridging the innate sensing of danger with the activation of the highly specific adaptive response. Now, beyond these major categories of lymphocytes and phagocytes, there are also a couple of other important immune cell types that play specific, really critical roles we should mention. First, mast cells. These cells are found throughout your connective tissues, but they're particularly concentrated near blood vessels and nerves, almost like tiny alarm bells positioned at strategic locations. Their key function involves a specific type of antibody called immunoglobulin E, or IgE. Mast cells have receptors that bind IgE molecules very tightly. Now, when an allergen, something like pollen, dust mite proteins, or certain food proteins binds to these IgE molecules already attached to the mast cell surface and specifically cross-links them, it triggers the mast cell to rapidly degranulate. Degranulation means they release the potent pharmacologically active substances stored in their granules, the most famous being histamine, but also others like heparin and various enzymes and cytokines. These substances are the primary mediators of immediate inflammation and allergic reactions. They cause blood vessels to leak, smooth muscle to contract, mucus secretion to increase, basically explaining many of the common allergy symptoms you might experience, like sneezing, itching, swelling, hives, or even the dangerous symptoms of anaphylaxis. They're the reason your nose runs during hay fever. And finally, let's loop back briefly to plasma cells. We mentioned them when talking about B cells. Plasma cells are terminally differentiated B cells. That means they've reached their final, highly specialized form. They're optimized for one incredible purpose. Their sole and specialized function is to produce and secrete massive amounts of immunoglobulins antibodies. They're easily recognizable under a microscope because they have abundant cytoplasm that is incredibly rich in a cellular structure called the endoplasmic reticulum. This extensive internal machinery reflects their immense synthetic capacity. They're essentially antibody factories, tirelessly churning out millions upon millions of these protective antibody molecules per second. These antibodies then circulate throughout the body via blood and lymph to find and neutralize their specific targets.
Wow. Okay, so after going through all these organs, the marrow, the thymus, the lymph nodes, spleen, MAPT, and all these incredibly specialized cells, B cells, T cells of different kinds, NK cells, neutrophils, macrophages, dendritic cells, mast cells, plasma cells, what does all this mean for us, the listener, just navigating our everyday lives? This clearly isn't just a static list of parts. It's this dynamic, incredibly interconnected symphony happening inside us, constantly working in concert. How does this intricate dance of all these different players actually manifest? Maybe you could walk us through a real-world scenario.
Absolutely. That's the key, isn't it? Seeing how it all fits together. The immune system is truly a beautiful and formally complex example of biological coordination and specialized teamwork. It's definitely not just a collection of individual parts working in isolation. It's a highly integrated network that ensures comprehensive, targeted, and, importantly, memory-driven protection against an almost infinite array of potential threats. So let's trace a couple of paths, maybe, to really understand this intricate dance in action. Okay, scenario one. Imagine you get a small cut on your finger while cooking. Common everyday event, right? What happens almost instantly? First, obviously your body's physical barrier, the skin, is breached. This immediately triggers your innate immune system right there locally. Within minutes, the local blood vessels near the cut expand and become leakier. This brings more blood flow to the area. That's the redness and warmth you often see, part of the inflammatory response. This increased blood flow also carries the very first cellular responders, those speedy neutrophils we discussed. They actually squeeze out between the cells of the blood vessel walls and rush towards the site of the cut, guided by chemical signals released by damaged cells and any bacteria that got in. These neutrophils immediately start engulfing any bacteria that entered the wound. They're the initial cleanup crew, trying to contain the breach. They're quickly joined by resident macrophages, those big eaters that were already present in the tissue nearby. They also start phagocytosing bacteria, as well as clearing up dead cells and debris from the entry. Now, while this immediate innate response is busy holding the line, it's also actively sending out crucial distress signals, those cytokines we talked about. These signals alert the wider immune system, including the adaptive arm. As macrophages and critically those dendritic cells arrive at the scene or are already there, they don't just eat the invaders. They act as vital intelligence gatherers. They gobble up fragments of the bacteria, process them internally, and then those dendritic cells, acting as messengers, actually start to migrate. They leave the site of the cut finger, travel through the lymphatic vessels draining that area, and eventually arrive at the nearest lymph node, probably one in your armpit.
Wow, so the intelligence from just a tiny cut on your finger actually travels all the way up to your armpit lymph node. That's quite a journey for such a seemingly small event. What happens once that intelligence, carried by the dendritic cell, arrives at the lymph node checkpoint?
It's an incredible journey, isn't it? And it highlights how connected the system is. Once those dendritic cells arrive at the lymph node, they become the primary presenters of the bacterial fragments, those mugshots of the invader, to the huge populations of T cells and B cells waiting there. In the T cell areas of the lymph node, the dendritic cells present the antigen specifically to naive T helper cells, the CD4 plus ones, that happen to have the right T cell receptor to recognize that particular fragment. These specific T helper cells then get activated. They start to multiply rapidly, clonal expansion, becoming the conductors for the rest of the specific immune response against that bacterium. They then help to activate B cells within the same lymph node that also happen to recognize fragments of the same bacterium via their B cell receptors. These activated B cells then proliferate and differentiate. Some become those plasma cells, the antibody factories, which start churning out millions of specific antibodies tailored precisely to that invading bacterium. Simultaneously, depending on the threat, some T helper cells might activate T cytotoxic cells, the CD8 plus ones, although that's more common for intracellular pathogens like viruses. These newly activated effector T and B cells, plasma cells, then leave the lymph node, enter the bloodstream, and travel back to the site of the infection in your finger. The antibodies circulate, tagging bacteria for destruction by phagocytes like neutrophils and macrophages. If needed, cytotoxic T cells would eliminate any host cells that got infected. Now, this whole intricate process, from the cut to the full adaptive response kicking in, it takes a few days. That's why your cut might get a bit swollen, red, maybe a bit sore before it really starts to heal properly. But the magic, as we said, doesn't end there. As the infection is successfully cleared, a small population of long-lived memory B cells and memory T cells, specific to that bacterium, is left behind. This is the aha moment of immune memory in action. Should you encounter that exact same bacterium again in the future, maybe from another cut, these memory cells are primed and ready. They will launch an immediate, supercharged, and highly effective response, often clearing the infection before you even realize you were exposed or feel any symptoms at all. This is why you rarely get the exact same strain of strep throat twice, for example.
That's a perfect example, yeah. Seeing how the immediate innate response contains things and then calls in the highly specific, powerful, and memorable adaptive response, it really shows them working together seamlessly. Okay, what about a different kind of threat? Maybe something like a virus, perhaps the common cold or flu virus entering through a respiratory system. How does the dance play out there?
Another excellent scenario, and it highlights slightly different aspects, particularly the role of MALT and cytotoxic T cells. So, when a cold virus enters your body, let's say through inhalation into your nose or throat, that mucosa-associated lymphoid tissue, the MALT, lining your respiratory tract is immediately on alert. Cells like resident macrophages, and again those crucial dendritic cells in the airways, will quickly encounter the virus particles. These vigilant dendritic cells, acting as those frontline scouts, will internalize the virus, maybe pieces of it, process it, and then they begin their migration journey, this time to the local lymph nodes draining your respiratory tract, maybe those in your neck. They're carrying the viral blueprint. Once they arrive at the lymph node, the dendritic cells present fragments of the virus, again on MHC molecules, to specific naive T helper cells, and also interacting with B cells. This kicks off a similar cascade of events as with the bacteria, but tailored to a viral threat. The activated T helper cells will coordinate different units. Some will stimulate specific B cells to mature into plasma cells. These plasma cells will then produce antibodies specifically designed to neutralize that particular cold virus. These antibodies will circulate in your blood, but also importantly get secreted into your mucus lining the airways. There, they can bind to virus particles, preventing them from infecting new cells, and also tagging them for destruction by phagocytes. Simultaneously, this is really critical for viruses. T helper cells will also activate T cytotoxic cells, the CD8 plus killer T cells. Why are these so crucial here? Because viruses replicate inside your own cells, turning them into virus factories. Antibodies generally can't get inside cells to stop this. So the activated cytotoxic T cells will circulate, seek out, and then directly destroy your own body cells that have become infected with the virus, recognizing them by the viral fragments displayed on their MHC class I molecules. This stops the virus from replicating further by eliminating the factories. And here's where another layer might come in. Your natural killer, NK cells, could also be active very early on. If the virus tries to be sneaky and makes the infected cells hide their MHC class I markers to evade those cytotoxic T cells, the NK cells, remember, are looking for that absence of the self signal. They'll detect these stealthy infected cells and eliminate them immediately, acting as a crucial backup system. So this whole intricate dance involving multi-dendritic cell migration, T helper coordination, antibody production by plasma cells, direct killing by cytotoxic T cells, and surveillance by NK cells ensures a comprehensive and robust protection against the viral invader. It's truly keeping your body's integrity intact. And honestly, variations of these processes are happening inside you right now continuously, whether you're aware of it or not, dealing with minor challenges constantly.
What an absolutely incredible journey we've taken today. Really deep diving into this silent yet incredibly complex world of your immune system. From the bone marrow and the thymus, where these immune cells are born and then educated through that surprisingly rigorous selection process, only 2% making the grade, to the lymph nodes and the spleen, these filters and meeting points where they gather intelligence and actively engage threats, and then getting into the specialized soldiers themselves, the B cells, churning out those precision antibodies, the different kinds of T cells, coordinating the whole show and eliminating infected cells, the macrophages acting as both immediate responders and crucial communicators, NK cells looking for hidden threats, neutrophils rushing in, dendritic cells scouting, mast cells sounding alarms, plasma cells building the weapons, each playing their vital, distinct role in what really sounds like a dynamic, ongoing symphony of defense. We truly hope this deep dive has given you, our listener, a newfound appreciation for the incredible complexity, the elegance, and just the tireless dedication of your body's defense mechanisms. It's this continuous, incredibly sophisticated operation happening inside you right now, this very second, keeping you safe from countless dangers you probably never even perceive. Which I suppose raises an important question as we wrap up. Knowing just how exquisitely balanced and sophisticated this internal defense system is, what are the implications for us? What are the daily habits or maybe environmental factors that could subtly strengthen, or conversely, perhaps silently undermine its continuous, tireless work? Things we do every day. And, given its complexity, how might our ever-deepening understanding of this system lead to even more revolutionary breakthroughs in medicine? Not just for fighting infections, which is amazing enough, but maybe even for things like re-educating the immune system to more effectively tackle challenges like autoimmune diseases or allergies, or even making more progress in the ongoing battle against cancer. It feels like we've just scratched the surface of this fascinating world inside you today, and there's clearly always so much more to explore. Thank you so much for joining us on this deep dive into the structure and function of the immune system. Keep learning, keep questioning, and definitely stay curious.